Good morning or good afternoon, everyone, and welcome to this first NIA webinar of 2023. Today's topic is characterization, and this is the first installment in a two-part series that will see the second installment 1 week from now on Monday, 6th of February, same time. Before jumping right into the topic, I would like to take a moment to introduce you to NIA or refresh your memory if you're already familiar with who we are and what we do. NIA is an industry association based in Brussels, but with offices in the UK, Netherlands, and Switzerland. Our overall mission is to support the successful development and market rollout of nanomaterials. And in order to do so, we support our members with a number of key activities in order to go from the lab to market. We help them understand what kind of regulatory environment they need to comply with in order to place their products on the market. We support a conversation that enhances the public's understanding of how nanomaterials work to foster user confidence in their performance and safety. And because, as you know, nanomaterials have a broad range of potential applications, we also support companies and researchers with identifying what kind of challenges their materials are best placed to address and how they can reach out to potential customers. In order to do so, therefore, our activities are quite diverse. We are obviously involved in the development and implementation of a regulatory framework that is predictable and provides the right environment for researchers and companies to thrive in. We create visibility opportunities for our members so that they can reach out to interested partners, be that for research purposes or for commercialization of their products. And we try to do this with the global nano ecosystem in mind. So from research and innovation to the applications and market rollout. Because of that, our members are not just the companies that produce and use nanomaterials, but also the providers of those collateral products and services, such as lab equipment manufacturers and the providers of regulatory advice services, as well as research organizations and national and international associations. In terms of regulatory and policy topics, we try to follow regulation at the global level, and we follow, therefore, chemicals legislation and standards across the globe. We are also involved in the development of OECD testing guidelines, and we keep an eye on recent developments such as safe innovation, safe and sustainable by design, and advanced materials. We also try to advise our members on the relevant trade developments that can impact their business. And of course, we do not do all of this by ourselves, only with our members, but with continuous engagement of the relevant stakeholders in this ecosystem, which includes policymakers, but also other industry sectors, civil society organizations, and academia. Now, the webinar that you are participating in today is part of our webinar series, Nano in Action and Nano in Business, that looks at different examples of applications of nanomaterials, as well as relevant developments in this space. Also, as online activities, if you are interested in learning more, you can join our very broad and active LinkedIn community. And as we are moving back to more in-person events, you can keep an eye on the NIA events calendar to know what meetings and trade shows we will be attending, including with our Nano Pavilion, which is a member-exclusive benefit for NIA members to have an exhibit space at selected trade shows. For those of you more research-oriented, I also want to mention that we are involved in a number of cooperative projects, normally funded by the EU, on topics of relevance for nanomaterials, innovation, and safety. How to get involved? Of course, if you are attending this event, it is probably because you already receive our newsletter and or are involved in our LinkedIn community. But if that's not the case, it will only take you a few seconds to join. And of course, if you would like to do more than just receive information from NIA and you would like to actually be in the driving seat, we're very happy to discuss membership opportunity. As I mentioned, we have different membership categories. So I think that whatever your organization type, we probably have a membership formula that works for you. With that, I think we can go back to today's main topic. And before I hand over to our first speaker, just a couple of housekeeping reminders. We are recording this event to make it available on the NIA YouTube channel so that you will be able to catch up with it afterwards. All participants will receive the slides. And we will have a Q&A session at the end. So at that point, you can either type the question in the chat or raise a virtual hand and ask the question directly. Until then, I would ask you to ensure that your microphone is turned off. And with that, I will stop sharing my slides so that our first speaker of today can put his presentation on. And I will introduce him in the meantime. So Dr. Enrique Carbo is head of the IMIS facility at INL. IMIS stands for Advanced Electron Microscopy, Imaging and Spectroscopy, which I'm very happy I managed to pronounce in one breath. And Enrique, whenever you are ready to put your slides on the screen, the floor is yours. Thank you. And I think you might also want to unmute yourself.
Okay. Good afternoon.
Perfect.
And thank you, Chiara, for the nice introduction and for the invitation to be here today and present and do this presentation about electron microscopy. Let me share. Okay. Can you see the screen already, right?
Yes, thank you.
Okay. Okay, thank you. Okay, in the next 15 minutes, the idea of this presentation is to show you an overview of the capabilities of electron microscopy techniques for the characterization of different materials at the nanoscale. As Pierre already said, I'm the head of the electron microscopy facility here at International Iberian Nanotechnology Laboratory, who is a quite new research institution located in the north of Portugal, north to the city of Porto, and is a lab that was founded in 2008. It's an intergovernmental laboratory between Spain and Portugal. And we have approximately 350 researchers from many different countries. These are our facilities here in the city of Braga. And basically, the research we carry out here is divided in 6 clusters. We have groups working in sustainable environment, others in health, smart and digital nanosystems, advanced materials and computing, food, and clean energy. And then we have the research core facilities and the engineering groups that provide support and service to the groups here at INL. The electron microscopy and spectroscopy facility belongs to the research core facilities, and we also provide services for external users, academia and industries. Our facility, our electron microscopy and spectroscopy facility is certified in quality. So the scope of this talk is showing this slide. I will give you a brief explanation about why electrons are useful to characterize samples. A brief history of electron microscopy, and then I will show you the main difference between scanning electron microscopy and transmission electron microscopy, how these two types of microscopes work. And then some examples of the capabilities we have here at INL for the characterization of these materials. So all this history of electron microscopy starts around 1931. The origin of everything comes from this group of researchers, basically in the double nature or dual nature of the electrons, that electrons, as you know, can behave as waves or as particles. And this was initially a hypothesis and experiment that Thomson, Reid, Davidson, and Gerner did. They show that the electrons could be diffracted when passing through thin crystals of nickel. So they act as waves because they have diffraction pattern. And also Louis de Broglie in his PhD thesis in 1924 proposed this hypothesis that electrons can behave as particles, and waves. And this is very important because this is the idea that Ruska and Knoll mainly used to develop the first electron microscope. The first electron microscope came to the idea of the first electron microscope came because the classical optical microscopes reached their limit because we cannot characterize or visualize samples with a resolution below of the wavelength of the light. Wavelength of light is in the range of hundreds of nanometers, and considering the diffraction limit established by Ernst Abbe, we cannot go below 200 nanometers. So this is the main limitation of the optical microscope. So considering that the electrons can behave as waves, Ruska proposed to use accelerated electrons as source to characterize materials, and this was feasible and was possible. They built the first electron microscope in 1931, and they already reached resolution in the range of tens of nanometers, so much better than the maximum resolution that the optical microscopes can reach at that time. So basically, how TEM or SEM works. TEM is the acronym of transmission electron microscope, SEM scanning electron microscope. How they work. Okay, this is an example of a scanning electron microscope. So what we have is a source of electrons on top. All these machines are in ultra high vacuum, the column and the chamber of samples. We have a source of electrons on top. Then these electrons are accelerated, in the case of the SEM, between 1 and 30 kilo electron volts. And once we accelerate the electrons, then we have several electromagnetic lenses to generate a probe, an electronic probe on top of our sample. Here we have, at the bottom, we have our sample. In our case, it can be a biological sample or a material. And we have an electronic probe scanning the sample. This is the reason why we call it scanning electron microscope. What is the main features of this technique of scanning electron microscope? Basically, the resolution we can reach with this technique in terms of imaging resolution is 2 nanometers, even 1 nanometer. The sample must be conductive, but this is not a problem. We have sample preparation equipment to make the samples conductive, depositing a layer of metal or a layer of carbon on the sample. We can make it easily conductive, and the samples don't need any additional preparation. So we don't need thin samples or other specifications. So we basically can take a bulk sample, place it in the sample chamber, and scan the sample with the electron beam. The resolution, as I told you, is around 2 nanometers. And the information we can get from the image, from the sample in this case, can be an image, can be a spectrum, or can be a diffraction pattern. As you can see here, we can characterize different type of nanoparticles, catalysts, fibers, or even we can analyze cross sections of different materials. The different contrasts we can see here tell us that the sample has a different chemical composition, and this different chemical composition can be confirmed with spectroscopic techniques in the scanning electron microscope. So we can combine the imaging mode with the spectroscopic mode. As we can see here, this is an image of the SEM, and we can overlap this image with the spectra and determine that the different contrast comes from different composition. This area here on top is made of iron. Here we have chromium, and here nickel. So this is confirmed by the energy dispersive X-ray spectroscopy techniques. We can also get diffraction information that tell us if the sample is a single crystal or is a polycrystalline sample, the different orientations of the crystals in the sample. This information can be obtained by electron backscatter diffraction. So on the other side of the SEM, we have the TEM, transmission electron microscopy. The main difference between an SEM and a TEM is, as you can see, the mode as we analyze the sample. In an SEM, we have an electronic probe scanning the sample. The sample can be a bulk sample. We don't need thin samples. However, in a TEM, the sample is not at the bottom of the column. It's in the middle of the column because we need that the accelerated electrons go through the sample. So for that reason, we need very thin samples. Very thin samples refers to something below 100 nanometers. So here in TEM, the sample prep is a bit more difficult than in SEM. In SEM, we just need conductive samples, and this is something that we can get easily with a layer of carbon, with a layer of some metal. However, in TEM, we need thin samples, and for that, in case we don't have thin samples, if we have a bulk material, we have to do a previous sample prep to cut a piece of our sample and reduce the thickness to 100 nanometers or below. The advantage of the TEM is that the resolution is much better than in SEM, is at least 10 times better. The voltage we use is a bit higher. In the SEM, we talk about 1 to 30 kV. Here we talk about voltages from 60 to 300 kV. And also we have 3 different groups of techniques: imaging, spectroscopy, and diffraction. In imaging, we can get images of our samples to study morphology, particle size. In this case, we have nanoparticles of iron oxide, use it in MRI as a contrast agent. Here we have fibers used in catalyst. And here this is a covalent organic framework used to trap environmental pollutants. So we can get images of the different morphologies, sizes. This is a multi-layer film. And we can also combine these images with chemical analysis. So we can see here the different colors means that the areas of the sample has different chemical composition. This is a multi-layer sample with cobalt and strontium. These are particles made of silica and palladium catalyst, or a contrast agent made of gold and iron. So apart from that, we can also have structural information that comes from the crystalline structure of the sample by diffraction techniques. So basically, to summarize, what we can get from SEM or TEM is complementary information. Let's say SEM is a technique where we have a scan electron beam scanning the surface of the sample. We don't have any requirement in terms of thickness. Independently of the thickness of the sample, we can analyze it. We just need to make the sample conductive, and the resolution is 1 or 2 nanometers. However, in TEM, the sample thickness must be below 100 nanometers, so the sample prep is a bit more difficult, but the resolution and the information, structural information we get, has a better, we can go deeper in terms of imaging resolution, and we don't need conductive samples. This is the advantage. So some examples of these machines. These are the machines we have at INL. We have 4 TEMs, 2 SEMs. All of these machines are available for the community if you want to discuss or contact us to discuss this. the capabilities and your samples, you can do it. So here are some examples of the SEM analysis we can do. In this case, we have a SEM installed in a machine with a focus ion beam that allows not only to analyze the surface of the sample, but also to cut and make holes in the sample and do the analysis of the cross sections in case you have multi-layers or samples with different composition in depth. We can also combine this with the chemical analysis. Different colors here are showing the distribution of the elements along the cross-section of the material, or analyzing different phases. This is the cross-section of a material, showing each number, so the different material in the sample, different phases that we have. And this is SEM analysis of the cross-section. In comparison with this, we have the images that we can get in a TEM. As I showed before, also we can combine it with chemical analysis. And another advantage of the TEM is that if we have a corrected TEM, it means that we have an accessory in the microscope that is a corrector. We can get relatively easy atomic resolution. As you can see here, this is an iron oxide nanoparticle. And if we go a bit higher in magnification, we can see the atomic columns, the position of the atomic columns of iron, maybe are visible here. Each one of these spots here belongs to an atomic column of the iron. So with this technique, we can get a resolution of up to 60 picometers in the machines we currently have, 60 picometers in imaging, and also combine this, as I told you before, with chemical analysis. We can as well determine if the atomic column we are analyzing is made of, in this case, manganese, lutetium, or iron, as this is the case. And just to finish, the latest advances in electron microscopy are going in the direction of doing experiments in situ inside the microscope. As I told you before, the microscopes are in high vacuum or ultra high vacuum, but it's possible now with the new sample holder to reproduce the experiment with liquid, with temperature control, with gases, basically to simulate inside the microscope the working conditions of different materials. For example, if we want to analyze how the catalysts behave under working conditions, we can put a gas inside the chamber with a sample and analyze in situ how the catalysts behave. Or even we can put liquid inside the microscope and analyze in real time, and also with control of temperature, how the sample behaves. Even for battery materials, we can apply a voltage and see how the material behaves when we charge and discharge to see what is the origin of the degradation of the materials during charge-discharge cycles. So with that, I think is all I want to show. This 15 minutes, as I told you, all these machines are open for the whole community, for industries, academia. We have access to all of them 24 hours, 365 days per year. And if you want some additional information, I can share our brochure with all the capabilities of our machines. That's it. Thank you very much.
Thank you very much, Enrique. Although I do hope that availability 365 days a year doesn't mean that you'll have to work on Christmas if anyone needs urgent analysis. All right, I think if you could stop sharing your screen, that will make it possible for our next speaker to put her slides on the screen. And in the meantime, I will introduce her. So we will have a presentation from Dr. Annika Krause, who is responsible for internal coordination and management at PWB Analytics. and who is here in her capacity as a participant in the Nanopad project, of which PWV Analytics is leader of work package 2. So Annika, whenever you want, you can put your slides on the screen and unmute yourself.
Hello, everyone. Thank you for inviting us to this talk. Just a second, I'm going to share my screen.
Of course.
Just
Perfect.
let me know. Yeah,
we can see it now. Okay. Thank you.
All right. Thanks, everyone. So I'm going to present to you today a process analytical technology to characterize highly turbid materials. And namely, this is photon density wave spectroscopy. It can be used in a wide range of applications, for example, in chemistry, food industry, cosmetics, or in biotechnology or medicine. And a big advantage of this is that we can determine the scattering and the absorption characteristics independently with just one measurement. And then from the scattering characteristics, we can derive information about the particle droplets or cells, and with a bit of theoretical modeling, even the particle size. And from the absorption characteristics, we can then derive characteristics such as color, chemical composition, and temperature. This is how the instrument basically works. So we have laser light, which is guided through an optical fiber into the material. There it creates, through multiple scattering, the so-called photon density wave, which is again detected by a second optical fiber after a specific distance and then analyzed. And based on the intensity and phase difference between the emitting and the detected fiber, we can then derive the scattering and the absorption characteristics of the material. This is an example I just want to show you to stress the advantage that we don't need to dilute our samples before measuring. So here you can see the measured particle diameters of polymer lattices in different concentrations, and you can see that we can measure them fairly reliably up to volume fractions of 60%. And that's a big advantage compared over standard measurement methods where you usually can only operate in very low concentration fields, and that then requires dilution in many applications. And if you have to dilute your sample, then it's difficult to do this inline sensing, like the live monitoring of a process. And here we have an example of such a live monitoring of a process. So in red, you can see the droplet diameter of oil and water, like the oil droplets in a water emulsion. 20 volume percent we have here. And this is a phase inversion temperature emulsification. So we start off with large droplets in the macrometer range, and we end up in the nanometer range in the end, about 100 nanometers maybe. And you can really nicely see how we can observe this inversion live with PDW spectroscopy. And then just a few words about our company. It was founded in 2013, so we're going to celebrate our 10th anniversary this year. And it's a spin-off of the University of Potsdam, where this PDW spectroscopy was developed. And the aim of this foundation of our company was to be able to transfer this PDW technology from the research environment into the so-called real world, so into industrial applications. And we're currently, as already mentioned, members of the Nanopad, or participants in the Nanopad project, which is funded by Horizon 2020. And yeah, I can only stress that we're having a great team. It's really fun and rewarding to work at PDW Analytics and push this technology further and try to transfer it into industrial applications. And feel free to contact us either later in the Q&A session or also via mail. Here, I want to say that you can, of course, contact us at PDW Analytics. Here, we are still looking into research and development, but more on the commercial side. But there's also more applied and fundamental research and development going on at the University of Potsdam. So there are also other projects involved with PDW spectroscopy. And you're very welcome to check both out. And please just contact us whenever you have any questions. Thank you very much.
Many thanks, Annika. And I will now go directly to the next speaker affiliated with the NanoPad project. That would be Dr. James Bolton, who's a researcher with Iris Technology Solution. And I would ask James to share his slides whenever you are ready to. We can see the slides. We can't hear you though yet, so.
Right, can you hear me now?
Oh, yes, we can.
Okay, right. Okay, so we are Iris. We are a digital advanced engineering company specialized in photonics and artificial intelligence key enabling technologies for the digitization of industrial processes. We were founded in Barcelona in 2007. We have 65 staff. In terms of R&D investment, we've invested 45 million euros and done more than 100 projects. We have more than 15 patents that are active. Essentially, we are a provider of advanced engineering services, integrating turnkey solutions, all of which combine photonics and artificial intelligence to develop and integrate inline monitoring solutions with spectroscopy-based analysis. We are the manufacturers of the industrial-grade near-infrared analyzers under the Visum trademark, which I will present, and we develop data management platforms and AI decision support tools. So our expertise is in real-time monitoring, particularly in the food, pharmaceutical, and chemical industries, to improve their quality control and process standardization. So most of our devices are based upon infrared absorption spectroscopy, and these are integrated into manufacturing lines for real-time analysis of the products as they're being produced. Additionally, we develop chemometric models to extract information from the spectral data. We also do some machine learning and data fusion models for predictive control. Our product line, the first three products here are in the Visum line. On the left is the Visum Palms. That's a handheld portable device for determining the chemical composition of materials and mixtures. That works in real time. The second device is the Visum near-infrared inline, which does something similar, but that is to be used with a conveyor belt or a pipeline. And the third device is the Visum hyperspectral imaging. So that's a device that takes hyperspectral images of materials that are going on the conveyor belt and is used for analysis, detection and sorting of materials in pipelines. Our other two products, the Visum Raman inline is similar to the near-infrared Visum inline analyzer, except it's based upon Raman spectroscopy rather than infrared absorption. This one is to be integrated into pipelines with aqueous media and is used to measure the concentration of compounds in the pipelines. The final product is the Visum DeepSight, which is a machine vision system with deep learning for the analysis and detection of complex and indeterminate visible effects. So where we fit into the technical part is we are part of the NanoPat project, which is about online real-time characterization of nanoparticle production processes. It's an EU-funded project. And we are developing a device based on the principles of turbidity spectrometry to measure the size of nanoparticles in liquid suspensions. So turbidity spectrometry is based on the scattering of light by particles. This device can be used for nanoparticles from size 30 nanometers up to a few microns. The benefits of Iris's TUS device are the biggest benefit is it can be placed inside the particle reactor. So it provides real-time inline measurement of the particle size. It can be used for a wide range of concentrations or dilutions of the suspension. It also works with nanoparticles that don't scatter much light. The device is still able to collect enough signal. It can be used for nanoparticle suspensions such as polymers, silicon, and titanium dioxide. And finally, no control of the nanoparticle flow inside the reactor is required to use this device. My last slide is just a case study that we've actually done and implemented for a client. A client that is making multi-layer films, and prior to the involvement of Iris, the client was only able to measure the overall thickness of the 5 or so layers in the multi-layer film. We introduced our Visum inline device, and with that we were able to measure the thickness of each layer. The layers were of thickness 10 microns to 150 microns. So it's a bit bigger than nano-sized, but not too much bigger. And the benefits for the customer were an increase in production and less waste. The customer base for the project had paid for itself within 4 months. So if you'd like to know any more about our services, whether it's about the turbidity spectrometry device for nanoparticle characterization or anything else, please get in touch.
Many thanks, James. And I will now invite our last speaker for the NanoPat project to take the floor. I will briefly introduce him, Dr. Christian Hill, who is CEO and CTO at Brave Analytics. And whenever you are ready, Christian, you can go ahead. Thank you. I think you might have to unmute yourself.
Thank you.
Sorry, and start again. Sorry.
A big fail. I'm going to start over again. Thank you very much for having me. My name is Christian Hill. I'm going to present a new characterization technique called optofluidic force induction. And you can see my screen now. I'm the CEO and CTO of Brave Analytics. Brave Analytics is developing also within the NanoPat framework, the EU project, a new process and lab analytic device, optofluidic force induction. It's a brave new way in time-resolved particle characterization as a process analytical technology, but also can be used for lab. How optofluidic force induction works, what the new principles are, and how you can use it. This will be the topic within the next couple of minutes. So we are a spin-out, a startup in Graz in Austria. It's the second biggest city here in Austria. And we're coming from the Biophysics Institute at the Medical University of Graz, being in this beautiful building right there, having lots of research facilities available for reference methods and project developments. What we do, of course, the problem we solve is particle characterization. Thousands of tons of particles and solutions get produced for lots of different applications. They all have one in common. You need to know the different characteristics for those in order to really analyze and then refine and of course improve also your processes towards waste reduction, but also performance control. This is not something we say, this is also basically recognized by the European Commission, that modern particle research needs to have faster and real-time characterization for process feedback. How do we do it? We have this new approach called optofluidic force induction, OF2I. We combine biophotonics and microfluidics. We pump the fluid containing the particles through our measuring cell, and we introduce a laser. This laser has a couple tasks. It first makes the particles visible, but it also changes the trajectories. The trajectories are changed by photon momentum transfer as a new approach, and we watch the particles, how they flow through our measuring cell and how to scatter light. And all this can be used for continuous feedback, for real-time online nanoparticle characterization. And we do not need any Brownian motion anymore because we introduce the forces and the trajectories through the laser light. And this is done via the principles of the Nobel Prize winner in 2018, Mr. Ashkin, who studied optical forces on particles, and he discovered or invented the optical tweezer systems, which got him the Nobel Prize awarded. With this technology, we put this flow-through technology. We do not look at single particles, how to do it in optical tweezers. We put the same principle onto thousands of particles at the same time, and they can be flow through. So they're actively accelerated, and you can use this as a PAT device, and you can do this as a kind of a online characterization. Since concentration is always a topic, so we also developed a dilution system, or like a sample preparation, which is then directly connected to the process line. And the process line basically gets a little bit of sample into our device. Here, the correct concentration will be determined because we measure concentration there, and we give back particle concentration, particle size of a wide size range starting from roughly 20 nanometers up to a couple micrometers. Of course, it depends on the sample, but we also go into purity measurements, turbidity, and aggregates, adding spectroscopy approaches like Raman spectroscopy on a particle kind of base. Our application areas, of course, is in process quality control, real-time release testing in pharmaceuticals. This is one of our goals, and then identification of critical process parameters and so on. How do we do this? Well, for example, here with one use case, with Fresenius Kabi, we work on high pressure homogenization processes to really look into the different homogenization steps to characterize diffusions. Here you can see, for example, that we can really deliver concentration over size over time. And as soon as something changes here, for example, in this area, a processing step changed, and we directly can see this in a timely resolution every 10 to 15 seconds, you get the full number-based size distribution. For as a process feedback, we give so-called D values, and they are coupled back into their production line, and so we really can give the example, see something changing in the system as well. They can act on those signals. Before really going into production, sometimes it's very needed to also do R&D for upscaling devices, or even see particle formation as in lab. This is, we put the same technology, OF2I, into this lab device. And just to see one use case as well, we looked at protein RNA complexes, so-called liquid phase separation, which they form over time when you just put RNA to those proteins. And you can see here again time over size, and here on the color bar concentration, that after a while particles are forming, and we can really see them on counting based method, being here roughly 10 to the power of 5 particles each point. And when you go further and you put more RNA, for example, you see that after 5 minutes you really see a concentration and particles form. But you also can see on a very low concentration base here, maybe even single particle base, agglomerates forming in the size range of above 1.5 micrometers and so on. So this is just a small overview of optofluidic force induction. There's a whole team working on this. We are very excited for this new technology. We see very great advantages, of course, over other technologies, being a single particle base, very statistical value results in a very fast and timely manner. So we are going to keep on working on this. And if you're interested in talking to us, we are very happy to talk your application. Go with different particle system. We have experience with different ceramic particles, silicium oxide, silicium carbide, and so on and so on. Please just contact us. We can discuss your particle sizing range, what the online measurement means, and so on and so on. So far from me, thank you very much. This is Christian Hill from Brave Analytics, and we are developing online real-time nanocharacterization technologies. Thank you.
Thank you very much, Christian. I will put in a moment the link to the NanoPad project in the chat so that you can also have a look at what the project does in general and get a few more details. I would now like to open the Q&A session. So as I mentioned earlier, you can either type your question in the chat or just raise a virtual hand, and then you can go ahead and unmute yourself and ask the question. All right, in the meantime, because I see that a few people might be a bit shy or still digesting the information. So maybe I'll ask a general question to any of the speakers who would like to reply to this. When you talk with the organizations that contact you to use your services, are there any particular challenges that they encounter that you see come up with more frequency, or are there maybe some application sectors that have more challenges than others? I was thinking, I saw pharmaceuticals popping up quite a bit in the presentations, and I was thinking it's obviously one of the sectors with the most stringent regulations. So just as an open question, maybe we can go back to the same order as the speakers before, so we can start with Enrique and then go to the NanoPad speakers if you have any thoughts on this. And then later on, I will read out loud the question that has been added in the chat in the meantime. Thanks.
Yeah, in our case, regarding these requirements, pharmaceutical industry and pharma in general, they push us to the limits of the technique, of the machines, and so on. And for that, we have quite a new microscope, precisely oriented to life science and health, because the electron microscopy technique can be applied to many different samples, but specifically for life science and health, we have purchased cryo TEM that meet the requirements of the pharma industry and nanobio companies, because it allows to characterize the samples in their native state, freezing the sample and keep the sample hydrated but frozen. So this is what we have done in this issue for trying to meet the requirements from the companies, in particular with pharma.
Thank you. I don't know if Annika has any additional thoughts on that.
Yeah, just like Enrique already said, there are always challenging, like technological challenges that might have to overcome. For example, if you go into biology, you want to have an autoclavable inline system. So inline always means that you have to implement it inside the reactor. So also, does the probe fit into, like, is the length of the probe correct and so on? Or sometimes some materials show too much or too little scattering at certain wavelengths, but we are trying, or we are developing systems which are quite flexible to that, so we can adapt best to each case study. But yeah, we usually optimize our instrument depending on each case, and there are a few challenges that we usually have to overcome before it works perfectly in line. Yeah.
If it was easy, everybody would want to do it, I suppose.
Yes.
So James and Christian, and then I have a question from the chat, and Blanca, I have seen your virtual raise hand. Will come to you in a second.
Yes, I mean, you know, we work with pharmaceutical industries. Our devices are to be used in line in the manufacturing process. So we go through a sort of a set of question and answers of just to assure the client that the device we'll use will not interfere with the process, will not lead to too much energy being transported into the process, will not lead to any impurifications. So we would adapt the solution for the client, basically.
Yeah. Thank you.
May I add a little bit?
Of course.
One more aspect. Our background, we are coming from a biophysics institute in nanomedicine, and there, of course, it is easier to apply new techniques if you are in R&D. Now we are translating really into industry, and one thing coming up is, yeah, standardization and validation of systems. This is one thing we are facing right now that we really have to go when we go into, yeah, pharmaceuticals. And we're happy that we are about to do this also for this new technology. And yeah, so new techs are hard to implement when they're not validated. So this is the one thing, but there is a nice way to do this, and this is what I wanted to add.
Thank you, absolutely. Okay, I'll just read the question on the chat on can any of these techniques also measure nanosurface area in suspension? I see.
Well,
yeah.
if no one says anything, I would start. Since we are a counting method and we count particles and measure their size, we are of course also able to say how many the surface area of those particles. This said, we do not see primary particles. So if there is surface area basically hidden into pores or into different agglomeration, aggregation, and so on, then of course you need some absorption or some other things. But in general, if it's clear what your particle looks like, if you have a solution and that you have, like, for example, globally around spheres, then yes, it is possible to calculate surface area. In all the other cases, it's a bit more tricky, but we can definitely discuss this application, and I'm happy to maybe get in contact with you.
Then I would go to Blanca. If you want to ask your question or questions, you can go ahead.
Yeah, thank you very much. This is really interesting. I was thinking about safe and sustainable by design and how this could really help implementing this strategy, because I think you're already anticipating any kind of issues already at the pipeline before the product is ready. I was also wondering how do you do for quality control of processes? How have you started addressing this? I'm sorry, addressing the three presentations from Nanopad. Thank you.
Do you mean like whether we've already used this technology to improve the quality of a process or the efficiency of a process?
I think if you want to go into industry, you will have to, there are some ISOs out there, etc. If you already have been looking into how to comply with that, if they fit your very novel processes.
Well, if it comes to security and safety, we have the CE marking declaration, and for PDW spectroscopy is currently pretty simple, since the instrument is kind of can be put very far away from the probe. So the instrument where all the mechanics and the electronics are in can be actually stored in a separate room if necessary, if you have an ATEX environment or whatever, like a more complicated environment in the industry. And the probe itself is basically just metal and optical fiber, so there are no mechanical parts or electronical parts. So that makes it currently fairly easy to have a safety assessment, if that answers your question. I'm not sure.
Yes, that was my question. Yes.
Okay.
How about the other ones? Anybody else like to step in?
Yeah, I mean, I think you're also asking, have we done industrial trials with our devices at industrial partners, right? So the answer is yes, we're in that. We've done some trials and those are continuing.
Maybe I also will give a short answer here. So we are also implementing safety standards, safety procedures, like a lab device, and then also, of course, laser, all this laser safety, because it's solely optical. And for the implementation in pilot lines or in lines itself, we are an online technique. So we have a little bypass going into our device. If there is a very hazard environment, if the bypass goes then outside of this hazard environment, then it should be okay. It will, of course, increase the lag time until we actually can see the sample in our device. But it can be helpful. So you're out of your hazard environment, and you can still see with this lag then the process itself. Other than that, I'm not sure what else I should say to this actually. Maybe we can talk later a little bit more and deeper.
Thank you very much. Thanks.
Thanks. I see a virtual hand raise. Devin, you can go ahead.
Yeah, hi. I would like to actually follow up with one question, actually one aspect which Annika brought up just before, and that is how you have maybe, for instance, through the implementation of your technique, maybe help manufacturers improve their yield or quality of product. And just from, let's say, from the business side, what kind of improvements your clients and customers have seen. I think this would be very interesting to just, briefly highlight what the learnings were. And because, very often, I think what isn't highlighted enough is your background and your expertise, which contributes to all of this, because it's phenomenal what you've built up as a sensor technology, as a system. But, really the added value is the expertise of each one of you and what you bring in for your clients. So maybe just to offer some maybe brief insight without going into confidentiality issues there, but just to show the icing on the cake, as a manner of speaking. Yeah, thanks.
Or Annika, do you want to start?
Okay, as you like. Well, the advantage of PDW spectroscopy is, as I already said, that we don't have to dilute and that it's inline. And therefore, we can monitor processes pretty much live, like in a sub-minute regime. And that helps to, for example, to determine when a process has actually finished. So often people just run their synthesis for a fixed number of time, maybe 8 hours or so, but actually the process would have been finished by like after 6 hours. And this obviously saves time and resources. And I want to give a little example for that. My favorite PDW story is the production of bioplastics via bacteria. And with PDW, we managed to to follow the growth of the bacteria as well as the growth of the bioplastic inside the bacteria. So the bacteria turn waste fats and so on, like waste organic products, into plastic. And with PDW, we could follow that up and also have indicators if the bacteria are under or overfed. And in the end, could also perfectly diagnose when the process has finished and a new batch could be started. So here you can really have, like, you have multiple leverages of optimizing this reaction. And I personally like this really a lot because it's bioplastics and it's biodegradable and so on. So yeah, that's just an anecdote, but Christian, go on.
Yeah, thank you. Of course, waste reduction is one of the really crucial things, and also, like, when do you really stop your process? And going really into the possibility to first go into an online or a PAT technology and eliminating this batch then sampling and having the quality control in the end of a process. This is, I think, a very big advantage. And we know from different customers, 5%, 2%, 7% of just batch waste is produced each and every year. So bringing in online technologies, I think this is, for all of us and for all those speakers here, is a big advantage. Second, I think more for our technology, we do have this counting method where we are really also sensitive for very big particles, agglomerates, on a very low concentration base. And aggregation behaviors are things which, especially in biopharmaceuticals and proteins and antigens and so on, are always a topic. And there we do see that, we get lots of interest. We get also into this kind of production and waste reduction again, that you can see right away, is the aggregation happening before a filling line and so on and so on. So those are things we are on the way to really also provide spectroscopic data. This is something we do on a research basis right now, but we are planning on implementing this into the process lines as well. And there we can then really determine, is the particle, what kind of crystalline state is the particle in, what kind of faulty materials maybe are or species of materials are in your kind of production stream. So impurities and so on, especially when you go into very different applications. We're not there yet, but we're looking forward to have partners to develop something like this with them. And yeah, this is basically the thing we are working on and excited about.
Can I just say for Iris, totally agree with Christian and Annika. The main advantage of our technology is the real-time inline monitoring, so you know the particle size as the reaction is going on. Perhaps the point I'd just emphasize about Iris's solution is it's based upon spectral data. Rather than a classical analysis of which peaks are there and using those to identify the compositions, we're using a slightly different model, a chemometric model, which allows us to get information from what can look like fairly noisy spectra, but we can get information on particles and compositions from that.
Thank you very much. I have a new question in the chat, so I will just read it. Can these techniques measure the size of gas materials, or do we need to first convert them into liquid or solid state? Again, whoever wants to jump in.
So if we want to keep the order, as far as I know, I don't think that PDW spectroscopy can work with gas materials. Because there wouldn't be enough scattering. Like, we need quite a high amount of scattering, so I don't think that would work. So far, at least not. I don't know. I'm not aware of any applications where we have done that.
Okay, I'll just go to Enrique briefly if he wants to quickly mention anything so that he doesn't feel neglected.
Regarding electron microscopy, we need solid samples. We can use gases or liquids, but just to analyze the solid in liquid or in gas, the interaction with the gases, the molecules of gas or in liquid media, but we need to analyze the solid particles or the solid material.
Yeah, makes sense. Thank you. So James and Christian, I guess if you could just say yes or no, and then I have another question, and then Blanca, I see your hand raised. We'll come back to you in a second.
Yeah, no, our technique cannot be used for gases, no.
We have not done it so far. The principle also works in gases, but we stayed in liquids in the future.
Okay, there's some nice homework there for all of you, I think. I have another question that it doesn't give a lot of further details, so I'll just read it as it is. How about nanosurface modifications on the bulk surface?
That sounds like a job for microscopy to me, no?
With combining microscopy and spectroscopy could be possible to analyze, basically, maybe depending on the kind of surface modification yields, that is electron energy loss spectroscopy technique could help, because you can analyze basically how the electrons lose energy when they pass through the sample. So this is quite sensitive to any change in the electronic configuration of the material. So any modification in the surface should modify the electronic configuration of the surface, and this can be measured in many cases by yields. That is a spectroscopic technique that can be used in a TEM.
Okay, thank you. And I think, Blanca, we can go back to you if you have another question.
Yes, a question for Enrique. I think he had one of his last slides. I think I understood that he can see how a material will change. If that's right, how do you mimic that on the microscope slide?
Maybe you refer to in situ experiments?
Yeah, in one of your last slides, you were talking that you can follow like a catalyst, how it will change.
Yeah, exactly. This is basically, in the latest years, some companies are developing quite a specific sample holders that allows to create a very thin space where we have or liquid or gas interacting with the sample. So needs to be a very thin cell, because the electrons has to pass through this cell, and allows to do in situ in terms of we can apply voltage to the sample and see how the sample behaves with the voltage, apply temperature. We can keep the sample in liquid and put electrodes there and analyze how the sample behaves from electrochemical point of view. We can also do, for example, simulate the conditions, the working conditions of catalysts with gas. So there are different in situ experiments we can do now in a transmission electron microscope, controlling these parameters, trying to simulate the working conditions of the different materials, catalysts, battery materials, electrodes for lithium-ion batteries, and even with control of temperature, with control of pressure of gases, is one of the trends now in electron microscopy.
Right, thanks. I have another question, or maybe some crazy idea that came to my mind. Now looking into food contact materials and leaching, I was wondering if any of these technologies will allow us to see how leaches are coming out from materials, for example, for a food contact application. Like, imagine you have some kind of water filters made of, I don't know, some graphene, and you are continuously passing water there. And one of the requirements from EFSA is to quantify if there is any of this nanomaterial leaching out from the filter. Would this be a use for any of these technologies?
Well, for PW spectroscopy, probably not, because we need fairly high scattering solutions, and you're probably talking about really small concentrations, because leaching, like once it's turbid and you can see it by eye, then you don't need the technology anymore to prove it. I guess this is definitely a case for Brave OF2I.
Yes. May I? Thank you for this question. This is exactly something we are doing because very low concentration and liquids which only have a few particles can be measured very accurately with the same principle. We do this, for example, with nanoplastics or plastic surfaces in general, that you see how much particles leave the surface and then are transported via liquid. This can be done with all different materials. Also, the question before that if we could see modification on nanosurfaces, so if particles are leaving a surface and then are transported via the liquid, we can detect them and also characterize them with the same precision with our normal technology. So water analytics, very low concentration of some in food applications to really determine what kind of materials in there. Also combining this via an ongoing project with ICP-MS, that we focus particles and then detect them before we put them into an ICP-MS. This is what we're working on right now. And there are lots of applications in this low concentration regime there. So it's a definitely yes for OF2I.
Thank you, Christian, because I think this is at least the example I gave could be an issue for some people developing food contact materials, for example. It comes to mind. So definitely, yeah, at some point, maybe, I don't know if you want to follow that line, but it could be also interesting to see how that fits there. But thank you very much for the answers.
In combination with spectroscopics, then this would be the ultimate thing.
Perfect.
Thank you. Thank you.
Thank you. I think we have time for last question. I see, Devin, your hand is raised, and then we can wrap up.
Yes, I just thought maybe, we could also maybe highlight for the 3 nanopad systems how easy it is actually to use them, and how accessible you are making your technology and data through your user interfaces, and as well as maybe just show different options, like for instance, if you could also rent and lease your devices, and maybe what kind of projects could be possible. Just a brief overview. I think that would help. Thanks.
Yeah, thank you very much. So with that, I think we're almost at the end of our time. So I think there's very little for me to do. Just a reminder that the second part of this webinar is next Monday, same time. The link is in the slides you will receive, but my colleague will also copy paste it in the chat. In case you haven't registered yet, you can do so. As mentioned, we will inform you when the recording of this session is available, and you will all receive the slides. And I think in the meantime, all there's left for me to do is to thank the speakers very much for your presentations, all the participants for taking the time to join us this afternoon or this morning, and on a Monday, what's more. So thank you very much, and I hope to see you all next week. And in the meantime, have a very good week.